В рамках задуманого спільного лекційно-семінарського курсу між трьома інститутами, тобто нашим імбігом, Інститутом мікробіології, Інститутом клітинної біології. Ідея в тому, що кожен раз будуть чергуватися інститути, аудиторії, відповідно спікери, і можна буде розповісти, поговорити там про свою роботу, про якісь методичні підходи, про якісь цікаві статті, думки. Ось, і будемо сподіватися, що це підвищить значно і нашу продуктивність, і наше знання, хто чим займається. Ось, і допоможе якісь знаходити цікаві для себе теми, навіть для спільних досліджень між трьома інститутами. Ось, а сьогодні у нас цікава лекція, і я дуже радий запросити Аліну. Вона сьогодні англійською мовою навіть нам прочитає про Integrative approach for data analysis in system biology. So, welcome, Alina.
Саша, де це ж поінтер? Hello, I'm really happy to see all of you here, and today I will tell you about the systems biology and what does it mean to integrate the data in the context of systems biology. This is a really obvious picture. Probably all the biology students know this, and we know about DNA double helix structure, but also Francis Crick formulated central dogma of molecular biology. So the DNA replicates, and then the information transcribed into RNA and translated into protein. And on all of these levels, we have to deal with particular type of information. And I want you to probably change a little bit your point of view on this data. And for example, and the very, very important thing, this central dogma was formulated in the way that the flow of the information is unidirectional, so you cannot go backwards. That's how it was formulated back then. And also, I don't need to explain to you this picture, and we know about base pairs, which are complementary, but if people from different fields of biology or from different fields of science look on this picture, they probably will have a different association. For example, me, I would probably want to implement, for example, reverse complement function. Given the string S, I want to obtain new string S dashed, which would be reverse of the first string and complement to it. And I want to implement the function which takes as input this string S and which gives us an output. And if you take a piece of paper and just a pen, probably it's really straightforward. You will start from the end of string S, and you will process the string letter by letter. So you start with A, then T, G, and you will write down this S dashed, also letter by letter. But if you want to express it a little bit more formalistically, for example, with the help of a diagram, so you can give it to a programmer and ask him to implement the function, it may look like this. Actually, nothing difficult. It's basically the same. So we have our input string S. We have empty string S dashed because we want to fill all the letters in this string, and we need a pointer. And here the pointer is I, and we want the pointer to start from this letter. So we want to start from the end of the string. That's why we assign the length of the string S to the pointer, so we can start from the end. And then on each step of the iteration, we would need to check whether whether we reached the start of this string, because we start from the end and go here, here, here until we reach the start of the string. So we check whether we reached the start. If no, we need to check the current letter. Then we check if it's A. If yes, okay, we had A here. So if it's A, we will proceed smoothly and just add a complementary letter to our string S. Then we decrease the pointer, so we want to move backwards. After that, we will just repeat all the steps. Then again, if it's A, no, it's not A. Then we proceed with T and so on and so on. So when you formulate it like this, it's probably not the usual way of thinking of a biologist. It's more close to computer science. But I just want you to change your mindset a little bit so we can proceed and dig a little bit into system biology. And I have a question for you. What could be changed in this diagram or added so we can ensure a fault tolerance of this system? For example, imagine that you want to develop a web application and you want user to enter his string S and get the output. What can be wrong? What should we change or add? Yes, exactly. So if it's not A, T, G, and C, we should output the wrong message. And thanks for the Ruslan. This is just a little bit of an exercise so we can communicate. And now, after training your brain, we can dig a little bit into history. And probably a lot of you heard about Human Genome Project. It started in 1990, and it took nearly 10 years to decipher the human DNA, and still one of the largest biological projects in the world, which gathered almost 20 universities from 7 countries, and it costed almost $3 billion, so it's very, very expensive. And the logical continuation of this project is ENCODE project, because actually Human Genome Project told us that we have nearly 20,000 protein coding genes, but it accounts only for 1.5% of human DNA. So some people even considered the rest of the DNA being a junk, but it's not like that. And ENCODE project told us that this non-coding DNA has a lot of different functions, but why did it take so long for a Human Genome Project to sequence the human DNA? And you can see it on this picture, because it started in 1990. And unfortunately, only in 2007, the Illumina introduced a new sequencing machine which made the speed of the sequence much, much faster. And it took them 10 years, but now you can do it in like days. So now we even entering in probably new era, because with this single cell RNA sequencing, or if you know this sequencing machine that looks like a flash drive, it is called nanopore MinION, and you can really stick it in your computer as a USB. So it doesn't have a very good accuracy, but it's I think it's a step to something new. And if we look on the cost of sequencing, remember that Human Genome Project costed almost 3 billion, so it's somewhere here. And nowadays, you can probably sequence it in a few $1,000, I mean one sample, but from what I know, there are some projects from BGI, Beijing Genome Institute. They established it with a few new companies, one from them in Poland, and they want to sequence human DNA for $600. So that's what we have nowadays. And it has led to generation a lot of different assays. If you are interested, you can go to this link and read. This is a blog post of well-known scientist in the bioinformatics and systems biology field. So he just writes down a name of a new assay whenever he spots it in the article. So we have to deal with the huge amount of different data sets. And in its way, it has led to development to the development of multi-omics approaches, so transcriptomics, proteomics, and so on. It's people used to study it within a different within the one type of data, but nowadays it's not the case. And also the question, if we have all these branches of science to study specific processes, what kind of science or branch of science should study the organism and the cell and the whole system? And probably the answer is obvious considering the name of the presentation about system biology. So it's, remember the first picture, it was the central dogma, and now we can talk about the extended central dogma. And here we have the flow of information in a lot of different directions. So our model became much more complex. We have, I don't know, reverse transcription, long non-coding RNA, post-translational modification, and so on. And so we no longer talk about, for example, transcriptome. We have to talk about epitranscriptome because we need to gather more and more parts of the system into one. And the cross of two independent timeline events has led to creation of systems biology. One is the development of next generation sequencing technologies, and the other one is development of mathematical and computational methods, starting from modeling leucoperon and brain neuron activities, signaling transduction. So only when all these two timelines crossed, the systems biology was born. But only in 2000, the first Institute of Systems Biology was founded, and it's formulated the Holy Trinity of systems biology, which is biology, computational science, and technology. So technology generates a lot of data, and it gives the data scientists ability to construct models which gives new insights and new hypotheses, which are then verified by experimental studies, which again produce new data. So we have a full cycle which never ends. So what is systems biology? The first of all is quantitative analysis, which is impossible without next generation sequencing. And usually in systems biology, the data analysis and modeling is prior to hypothesis formulation. Well, it usually goes together, but if I were to compare traditional molecular biology with the systems biology, this point would be different. And the most important thing is the system biology aims at understanding the behavior of the system as a whole. And systems biology study the properties of biological systems, and some of them are really self-explanatory, but I would like you to pay attention to emergency, which is pretty interesting, because it means that when the parts of the system combined, it leads to creation of new properties of the system which are not present in each part separately. So this is a very interesting concept, and if you want to go into modeling, you should take that into account. And probably the most widespread and basic methods of the systems biology is reverse engineering. Consider you have your data, which usually looks like a matrix with numbers. Then usually you will apply some mathematical methods or statistics, Bayesian inference, or just simple correlation. And in the end, you will get your model. So now finally we will proceed a little bit toward the understanding of integration, and I would say that actually integration can happen both on this level and on this level. So we can integrate data before the analysis, or we can integrate the model from the two different studies. So the two main efforts in the context of integrative analysis, which I just told you, is the data integration and model integration. And each of these things is really huge to explain. So today we will focus only on data integration within similar data types. And the first question probably, where to get this data? Usually, of course, you need to perform some experiment, but even if you cannot do that, like most of us are here in Ukraine, unfortunately, you still have a possibility to use publicly available data, for example, from such repositories as Gene Expression Omnibus, which contains a lot of different assays. I'm not sure if you're able to see, but here the most huge one is expression profiling by array, or just microarrays, gene expression microarrays. And you can see it still is the largest part of this huge public repository. And for example, here is the expression profiling by next generation sequencing or RNA-seq. And of course, it's trending now, and I expect that in a few years it will be as much big as probably this one. So let's focus only on gene expression data now. And here we have two typical data types from where you can get these gene expression profiles. One is a microarray, and another one is RNA-seq. What is a microarray? It is a chip which contain probes on its surface which should capture the target transcripts. And in the end, you will get a picture like this, where each dot has different color and different level of brightness. So the brighter the dot is, the higher the expression level of particular transcript because it hybridizes with the particular probes. So in the end, specific image recognition algorithm translate this picture into numbers. So like the most brightest spot will be assigned the highest, the biggest number. And in case of RNA-seq, we have a little bit different situation because we need to count reads, which we map to the reference genome or transcriptome. It depends on the type of experiment you want to do, and the more very simple explanation. So the more reads you have, the higher the expression level. But it has its own problems, which I'm not going to discuss this. I mean, to count this is not usually straightforward, but what is important in all these cases, we will have to deal with the matrix, with the numbers. So here, for example, in the column is gene names, and in the row are samples. So, for example, in this case, each sample will correspond to one microarray. And if we talk about data integration within the same data type, we have two cases. The first is meta-analysis, so the results of individual studies are combined. And the second one is merging, so we need to adjust our numbers in a certain way so they can be comparable between different experiments. And let's talk first about meta-analysis, but before, actually, if you have any questions right now, you can ask them, because you don't have to wait till the end, because you might forgot what you wanted to ask, so feel free to ask. And the most common situation is the two-class comparison, so where the objective is to identify differentially expressed genes between two well-known conditions. And differentially expressed means expressed significantly higher or lower between two conditions you want to study. And here, for example, this is actually the same, but higher numbers are depicted as the red dots and lesser with the blue ones. And we actually can spot some kind of pattern, like this area of genes are highly expressed in this condition, and this area is not so highly expressed in this condition. But you won't always get such kind of picture. Sometimes you won't be able to grab this correlation just by looking on the picture. So you need to apply some statistics in order to find that. And let's say you have the situation when you analyzed two data sets and you got two lists of differentially expressed genes, or you never did any analysis, you just took two articles and you extracted these differentially expressed genes from these articles, and you want to try to integrate the results of these two studies. What options do you have in that case? So these some of the typical techniques how to combine gene lists from different studies, and each of it had its advantages and disadvantages. And for example, let's talk about the most simple one. Any one of you can really easily implement that even just by taking a piece of paper and a pen. Well, probably this one is not very straightforward with a piece of paper, so I'm not covering how to find differentially expressed genes here, but I certainly can someone in the future if you're interested. But let's say we got these lists and we got a p-value of for each gene. So first of all, we need to filter the genes by p-value here, so we can leave only those differentially expressed genes that are significant. And then, for example, we have 10 different studies. Doesn't matter from the articles, from your own analysis. So we just simply count how much time we find particular gene in each of this list. And for example, we just establish some threshold, 5 out of 10 times I should find this gene in the list, and if I do, I will consider this gene as differentially expressed. So this is the example of meta-analysis, the example of how you can derive one piece of information from several different studies. And it's really not a very complicated technique, and many people use it. So maybe this will be useful for you as well. Then what about direct integration? What about comparing these matrices of numbers numerically? First of all, we will have to deal with the normalization of data within one study. So before we can integrate something, we need to be sure that we analyze the particular data set correctly. And for different types of data which came from different experiments, we will have to use different methods, different libraries for normalization, because for example, from this picture with microarrays, we will usually get integer numbers. I don't know how to explain it. So not real values, not like this. Like this. I mean like this. But here we are counting. We won't have real values. We won't have something like 1.5. We won't have that. And because of that, we will have to use different statistics for these two different experiments. Because here we are counting, and here we are not counting. We just get this. So if you're interested, you can read these two papers. We'll ask Alexandra to distribute the presentation, because it compares the different techniques for normalization for different data types. And next, if we're sure that we normalized the data set correctly within one study, we can actually go for a cross-experiment normalization. And in that case, we will have to deal with such thing as batch effect. So actually, it's just a technical variation which can be introduced, for example, because you did your experiments within a different time frame. For example, this one you did this week, and the next one you did like in a month. And it is enough to introduce this batch effect in your study. But what is more important, sometimes it can be introduced by something you cannot even predict. So your task will be to gather as much information about your data as possible, so you can try to see if it correlates with the date of experiment or with your library preparation techniques or something else. And I will show you a few practical examples of what does it mean within study normalization and between study normalization. So here you can see, so I took 3 different studies. Here you can see the identifiers from Gene Expression Omnibus. So each of you can download it and read the articles, see the raw values of the expression levels, so you can actually touch that data. It's not something private. And on the left, you see a principal component analysis plot where each dot represents one sample. So actually, per one sample, you will have something like, I don't know, 19,000 genes. But you cannot easily plot that. So there is a special technique which reduces the dimensionality of data, so you can just easily plot your sample on a 2-dimensional scale. And this data is from placenta study. So, and with placenta, we know that there are 3 trimesters, first, second, and third. And we know that actually they are different enough to be clustered apart. So the first trimester of pregnancy is much different from the second, which is much different from the third one. But here you see that these blue and green guys are clustered together. And I know that that they cluster it like that only because they come from the same study, from one of these. But first of all, to be able to plot something in a comparable way with a normal scale, usually you will try to log normalize your data. And on the right, there is a density plot where on the x-axis we have our intensity in a logarithmic value. So the 10 is the highest expression value of some particular probe on the surface of a microchip. So you can see that the distribution, so each line represents one sample. The same as here, each dot represents one sample, and here each line represents one sample. So as you can see, the distribution of the sample, the distributions are kind of different. But if you want to compare samples between each other, you will have to make them look the same. And as you can see, after log normalization, it didn't really help. So next step, you might want to try within study normalization with the special methods developed for microarray data normalization, which is called RMA. So again, you can easily use it because it is implemented as R package in our programming language. So we have now much better density plot. And what kind of underlying logic here? I will try to explain very simply. Actually, with the microarrays, you will have 2 different signals. First, it's your let's say expression signal, and the second signal is a noise which is introduced by particular technology, the way how you scan the chip, noise introduced by the background lightness, because even if you have spot on the microarray with some kind of brightness, some spots are just as they are not completely black, and we call this a background. We need to remove that so we can identify which genes are not actually expressed. So the idea behind of this RMA was to divide your signal into two different parts, each of which has different distribution. And that's why they were able to achieve this kind of normalization in comparison to just simple log normalization. But let's check this plot again. And what we can see, maybe you cannot, but again, these blue dots and the green one are clustered together only because they come from the same experiment. And now, finally, we are ready to try batch effect removal, so this between study normalization. And again, for this, you will have to use totally different methods, which was specifically designed for removing this batch effect. One very, very widely spread is combat, which is a function from SVA package, again from R. So now our picture looks much better. We can see that first, second, and third trimester are clustered apart. And what is also important, this method is perfectly applicable to RNA-seq data as well. So this is kind of universal method for batch effect removal, but if you want to use it, you will have to tell the function the name of your batches. So you will have to know beforehand that this part of samples are from batch A and this part of samples from batch B. There are also a number of methods which try to do it without this knowledge of batches, but I don't really use them because there is a high possibility that I will remove some biological variation, which I don't really want to do. And what I want to point out there is that here we know that first, second, and third trimester, they are very dissimilar, but you may study some type of cancer, and you may want to identify cancer subtypes. And you won't usually have such a great picture where you have all three clusters apart. So therefore, you won't be able to tell, for example, that these are batches. Therefore, you have to always gather all the metadata, all the all the clinical variables for each of your sample, so you can try to verify if they correlate with this clustering. And if the date of the experiment correlate with this clustering, most probably you just have a batch effect. Okay, to sum up, again, we have meta-analysis. So we analyze one study, then we analyze second study, and only after that we combine our results. And here we have a direct integration of data sets. So first of all, we integrate our matrices or whatever we have, and only after that we try to derive some result and analyze it. But the question is, what is better, and in what cases I should use one type of integration over the other type. A lot of text, but I will try to explain. So here is the comparison actually of two different microarray platforms. And each dot here represents one probe. And on this plot, we compared the microarray of the same platform. And on this plot, we compared the microarray of older version of chip and of newer version. And we can see that correlation is not so good. And in this case, the data comes from different biological samples. But even though we have a very, very, very good correlation. And the obvious question is if the technology, if the differences in microarrays design influences the correlation so much, why should we use this direct data integration? What if such kind of integration will just ruin our data? So actually the answer is not that obvious, and you need to perform separate study just to understand which method is better. And I will actually tell you only the conclusion. Some different people were interested in that, and many people tried to apply different types of comparisons, like will you get more differentially expressed genes or less? And apparently, even if we take different microarrays of different versions of a different platform, we will still have a gain in comparison with meta-analysis. Because first of all, at least in different cancer studies, it showed that it will give us an ability to get more differentially expressed genes. And if we compare it with RNA-seq technology, which is more advanced, still we have a gain. So we have a better correlation with RNA-seq. So, and the most important thing, sometimes for some particular cases, like for example, for our analysis with placenta, you cannot have a lot of samples, especially for example for the second trimester of pregnancy, because usually you can do an abortion on the first trimester, you are forbidden to do it on the second, and you will only get material after the delivery on the third trimester. So having the data from the second trimester is very, very rare. And for example, one study can contain 4 samples and the other study can contain 5 samples. So for us, it has a lot of meaning to try to combine it and to increase the statistical significance of the obtained result, because we will make the impact of individual biological difference lesser, and we will see something which is more correlated with disease or what we want to find. But I would also tell that actually meta-analysis is also nice, because not always you can get the raw values, the numbers, this matrix with numbers. You cannot always have that. And sometimes you will have to deal just with the article and with the reported list of differentially expressed genes. So it is also a nice technique. So as usual, it depends on what you have, what kind of data you have, and what would you want to get. So actually this is all. I think we should better leave some time for questions or discussion. So feel free to ask me something.
Can you please explain what's the density of data?
So again, when you get this, wait. This is from one of microarray studies, and these numbers, they are not log transformed. And if we apply logarithm function to each of these numbers, you will get something usually from I don't know, from 4 till 10. The highest probably value I happened to see was 15. Yeah, it's like a distribution, because you will normally expect a particular amount of highly expressed genes. Like you will have lesser amount of very highly expressed genes, a bigger amount of middle expressed genes, and lesser amount of low expressed genes. So it's just a distribution of gene, not gene here, it's probes intensity values. Because I didn't describe it here, but we need to perform a step where we annotate our probes to understand which probe are mapped to which gene. So here are probes.
Питання до матеріалу вашого, ваших перших, третій триместр. Це які були матеріали? На третій, це після пологового матеріалу? Або перший і другий? Що ж там, власне, за матеріали? Які дільник збирали?
Це було на прикладі...
Перший, другий, третій.
Просто я перше хочу сказати, що це на прикладі публічних даних, тобто не нашого експерименту. Я просто це спеціально зробила для цієї доповіді, щоб проілюструвати. Зазвичай це перший триместр абортивний матеріал, плюс там ще треба дивитись на тканину, тут це хоріон. Другий триместр це була chorionic villus sampling. І третій триместр – це просто normal delivery. Так, післяпологовий, uncomplicated normal delivery.
Особливо цікавий третій триместр, його можна розбити, принаймні на 3 на 4.
Так, так і є, так і є.
Другий, там, більш-менш однаково, перший дуже такий різноманітний, тому що є закладки, так от чому ви називаєте вагітності третій триместр, якщо ви післяпологовий? Там же такі надзвичайні зміни відбуваються вже напередодні.
Мабуть, коректніше, так як ви кажете, просто дійсно, якщо дивитись окремо на третій триместр, там може бути different gestational age, і не завжди це дивлячись, що ми вивчаємо. Якщо це просто ми хочемо подивитись на нормальну пройдену вагітність, а інколи це буде передчасна вагітність, і там же просто цікаво дивитись тільки на оці, скажімо так, давайте це назвемо біологічний матеріал, який більше певного gestational age. Тобто це дійсно не дуже правильно називати так.
Але доскорінювали і літературні дані, так само, щоб потім нормалізувати за однаковими умовами, то вони так само описували цей матеріал і показували, на якій стадії вони брали. Що коли дуже важливо, коли ви робите метааналіз, щоб ті саме терміни були однакові.
Так, я думаю, до речі, може з цього приводу буде просто окрема доповідь, тому що ми цілеспрямовано займаємося таким дуже кропітким підходом до метаданих, які, на жаль, не завжди ті, хто завантажують такі дані, вони не завжди їх нормально описують. Є, наприклад, окремі дослідження, які визначали стать дитини за профілями генної експресії, тому що хтось просто не написав це. Тому що у них була частина даних, в яких було прописано, а частину даних вони там... треба бути дуже обережним насправді. Але чим більше даних ти маєш, чим більше маєш опису конкретного семплу, тим краще. Все. Дуже вам дякую. Сподіваюсь, якщо у вас виникнуть якісь питання після, бо насправді не завжди можна запитати під час доповіді. Може, щось там через тиждень виникне, то прошу, пишіть листи.
Можна буде тоді презентацію ось там мейл.
Ну там та скільки ж її? О,